Hmm. One little snowflake.
No, I saw more than that. I totally did.
There are more than that.
Come on.
John, we totally got some
Way a
stuff.
bit more than that went by.
Yep.
Yeah, it's going to be all stuck to the filter.
Yeah. All right, Andrew, is this enough for you guys? Which was scraped kind of, we think that it's on the filter, but what's kind of got off the rock there? Oh, so I didn't hear you.
Yeah.
I just said, hold on, I'm muted, which really doesn't make any sense.
I can also go for the front of the rock if you want more, in case you're worried about sampling.
So you think it's just stuck on the filter, which is why we're not seeing in the...
Yeah, probably. We can try maybe jostling the jars back and forth, one. And then kind of will hopefully knock some from the filter.
Yeah, it seems like you've scraped quite a bit of it, so...
Yeah, it looks like another tiny little strip in there too. They look at every sample you guys bring.
All right. Oh, yeah. Snowballs.
Okay.
Cool. Is that enough for you?
Yep.
Okay.
So this is a sponge that encapsulated this
Come
rock?
wide out.
Yeah,
Wow.
that's what it seems like. Abby says that Clathria mima is a tropical shallow example of a sponge that looks like this. The encrusting tunicates don't usually have those vein shapes on them.
Susie, would you please bring your mic closer to your mouth, please? They're low gain mics, so they got to be close.
Sure.
Thanks. Ooh, you're still kind of soft.
Cool.
Okay.
That was fun.
It was.
Scraping brains.
So I think we made a mistake. We really should have made it into a sugarhead skull.
Yeah. Little artistry.
Guys, we could do pumpkin carvings any day of the week. Subsea pumpkin carving.
Yeah.
All
so
right.
in the discussion of what this potentially might be, Carrie adds, we have collected thin encrusting tunicates in South Africa, which are very friable like that. I guess the texture is similar.
Friable.
Friable.
Sure thing. Yeah, that sounds great.
But Abby makes a good point about the veins.
Oops, overshot that.
Susie, everyone sounds good to you, right? The levels are good for everybody else?
Yeah.
You. Okay.
Roger.
The pumpkin carving comment made me think of something we did at Halloween with the vehicle, carved pumpkins, and we took them down in the bio box to set them on the seafloor. We opened the bio box. Nobody told us pumpkins float.
Okay, so
that's wonderful.
So we're right over the pick here. Can we sort of look to the right and look to the left?
Of course. Sorry.
Actually, if we look to the right, there looks to be a rock that looks like the other rocks.
You want me to look at that rock, or?
Yeah, can we zoom, look in on that rock, this one?
Yep. All right, Al, go ahead. You want to push on in there, Al? Yeah. I'm going to do one quick pop down.
Let me know if you want to go tighter.
Yeah, a little tighter and a little to the right would be great for me.
Oof. Sorry, one sec.
I'm holding.
Thank you.
That's beautiful. Can we just hold the shot for about 20 seconds or so?
Oh, sure.
I've got more lens too if you want it.
No, this is perfect. Showing exactly what I want. Those are, I think they're hydroids, but they're quite, it's like little palm trees on top.
Oh, yeah.
All good there, Andrew?
Yep, all good there. Thank you.
Okay, go ahead and come wide there, Al. All right.
We continue looking to the right just a little bit more, and then I think head down slope past the other pick.
Looks like there's a bunch of large bivalves here, right?
Where do you see those?
Jess,
Large clams.
would you like me to go in past the push course?
Yeah, sure. That's great, thank you.
Huh.
What you thinking, Andrew?
I just don't really recognize those at all.
Anything in particular you want to look at, the clams?
Yeah, can you zoom in on the clams, please?
Yep.
Actually, also like to look at this right here.
Sure. I'll turn my head so that we don't get the cores in it. All right, go ahead, Al. I'm gonna just do one jostle down. So you want to see this area first?
That's a microbial mat.
Yeah. Okay. So there's a
Shall I go in more?
Sure. It's the mat you guys want to see, or this rock in front of it?
The mat, although that's a cute little sponge on the rock.
Okay, I'm gonna do one pop up.
Yep.
Yeah, we know how that goes.
Yeah, for sure.
Too zoomed.
I'm gonna pop down. All right, go ahead, Al.
It's all the way in.
Sure thing.
Good shot.
Great. Can we try to stick a core in there?
Yeah, sure. You want to see these clams too before we do?
Yeah.
All right, Al, you want to come wide? And I'll center us up on the clams.
They're big.
Yeah. All right, Al, go ahead.
Weird. Those do ring a bell. Is that a little? No, that's a different kind of tube worm.
All good there?
Yep, all good. Let's see if we can stick a core. Low expectations.
All right, go ahead and come wide.
And if not, then we'll go rejoin the transect for sponges and corals.
Sounds good.
It's a shiny little critter swimming around this area too.
What was that? Is that like a young sablefish?
Maybe.
I think so. And there's this, I don't know if it's
Little
what the
soul over
silverfish is that's jumping around.
Oh yeah. There's
right.
also the soul.
That sole looks like it's already been like prepped for cooking.
Soul on the rocks.
Didn't like that.
Oh, well.
under. Ooh, more shadows.
I hope we're not supposed to keep the science chat serious all the time.
We appreciate the silliness of it sometimes too.
Yeah, it's been fun.
Erica says that what we thought was a small sablefish was actually a big hake.
Sorry? All right. This one here?
Yeah, that one there.
All right, let's see if it goes in.
I just want to telestrate for no reason other than that one.
Yeah, all right, bullseye. Oh, more bubbles, but definitely nothing.
I didn't see anything in Norbit.
Nothing?
Nothing.
Good check.
Yeah, let me see if I get more bubbles. You want to check some more?
Yeah, looking.
Okay. Can you dance around on the seafloor surface?
Oh,
And
no,
we're going to put Norbit on.
Yeah, okay. Well, obviously there's no core here, but
We learned something else.
learned that Norbit.
See a little bit in Norbit?
You did?
Yeah, just a little bit.
Okay. Andrew, are you sure you're not seeing things there now?
Oh my gosh, I'm not sure.
Nice.
You actually see sort of a dragon flying in Norbit at the minute.
Interesting.
It's very pretty.
Cool.
Oh.
Well, thank you. That was instructive.
Yeah. If nothing else, we had fun.
I know, right? Exactly. I think that's good actually there. Perched on a dime. All right.
Okay, so I think the next plan is to essentially come back down the slope and rejoin the transect. If it doesn't add much time to swing by those other, by those other two picks, that'd be great. But otherwise, I think we can just try to, or at least one of the picks.
Yeah, let's just get an overview.
Yeah.
I think we could probably go that way. Well, we won't go fast like that noise implied. We'll go really slowly.
So how much farther down is there?
That's where their point is. I think that's the next one, 3.04.
So,
Maybe they want to skip that?
Jenny and Meredith, would it be better to just go straight down the hill back to your transect, or to cut over to point 3.04 from where we're going, which will leave a little bit off of it? Jenny can't see the teleprompter.
It's really hard to see these guys. I can turn off the background map.
Wait, Al, can you put it on camera number, camera number 3?
Channel number 3, okay.
Channel 3, yeah.
All right, so I'm going to turn off the background map first. We are up.
There we go.
Yep.
Here. You guys, they came up past 303 and then came up to where we were, and then I believe these would be the next set kind of along this contour. I could zoom out if you want to see further along.
Okay.
Okay, she said she was thinking going towards 3.05, so
Okay.
We could just go straight past the pick to 3.05.
Well, it's going to be sketchy, but we can head in that. Well, we're going to need advisement from Jess because it's really all these ridges.
Yeah.
Stand by.
Would it be better to go straight down and then over?
Sometimes that's easier. We can also do like a nice kind of angle along the contour this way, Aaron. So it's like not like a dramatic.
So not straight, like you want to go this way?
Yeah, more like a 135 or 150, something like that.
All right, and then these are going to all be ridges, I think.
Yeah.
And then 3.05 is there.
Yeah, so maybe like 135.
Maybe if we could just have delay tactics for the next 11 minutes. All right, 135.
Yeah, I think so, right? As long as we pull art.
Yeah, let me check. 135. That's 135. Can you see that line?
And then along the contour after that.
And then you trape it along the contour. Yeah, we can have it be 135 over that will, or 120 over that pick. Is that okay?
Yeah, that works.
And then we'll go along a contour. We could maybe like contour down.
Yeah, I like that.
Okay. 4, 7 minutes. Ready to go. Okay.
If anything, it'd be, since they went up that ridge, we can go down the next ridge over, you know what I mean?
Yeah. All right, let's go to there, 118. And
118.
We'll go over.
Roger.
Okay, we have a plan. We're going to go down a ridge we haven't gone down and then cut over, is my understanding.
It's going to be kind of a valley, and then we'll jump over ridges.
I'm going to sort out my tether wrap issue.
Adventurously.
Yeah, I think we go to there, scoot over, and then wherever the ridge is in this craziness, they can go down.
Yeah, I think that sounds good.
Okay. And I forgot the number I said again. 117. And I've been talking to Bridge the whole time. Sorry, Bridge. Ready for a move?
Yep. All good.
Bridge and F. We got 10 meter step 117.
Big long nose skate.
Yeah.
Pretty rock.
All good with me? Yeah, thank you. So that pick, it's about 60 meters, 70?
A lot, 153.
Oh, wow. Brain way off in this calibration mode.
Bridge nav, another step, same bearing.
Oh, I see.
Yeah, it was very left.
There's a hagfish.
It's the last few minutes of our watch.
Last watch. Bridge nav, another step, same bearing.
Is that a microbial mountain or is that a...
Yeah, that's what I thought it was.
Down here and to the left?
Yeah.
Yeah. Hard to tell.
Yeah, you'd have to poke it.
Do you want to zoom or do you want to poke?
Yeah, can we just zoom?
Yeah, go ahead. I'll go ahead and zoom.
I'm zooming.
That's great, yeah.
Rock.
Let me know if you want to go farther.
Rock.
Rock. Nope, that's good. Thank you.
All right, go ahead and come mine.
Well, those depressions without a big rock in the middle.
Yeah. I feel like we should move a rock into the depression since we took one out.
Maybe that box grab pushed it out when it was scratching its back.
Bridge nav, another step, same bearing.
What was that, Bob? Thanks for the watch.
Bob's here first. What's that about?
Something on the right in that rock.
All right, I'm signing off.
Thanks for a great shift and a great cruise, guys.
Yeah, I'll second that. Thanks, everybody.
You're certainly welcome. On behalf of Jess, who is not online at the moment.
Meredith, Liz, let us know when you guys are ready to sign into VLink. We'll sign out. Thanks, Andrew, for letting us tag along to all these fun seeps. I learned a lot this week. All right, and thanks, Nautilus. Signing out.
Yeah, this is Kurt signing out too. Thanks, everybody.
Hello, this is Meredith Everett
I am logged in.
Yep, this is Liz. Can you hear me?
I can hear you loud and clear, Liz. So just like us, the ROV crew is currently in the midst of a watch change. They're handing off to the next team who will be driving us around for the next 4 hours, and at the end of which they'll be recovering the vehicle this evening.
So, Meredith, I think they're headed downslope right now. I'm trying to
Yes.
see. And I think, so if they're going to proceed, hello?
Yes.
Oh, I was thinking we could, it seems to be repeat. Okay.
Just cut out. Oh, make sure all your videos are muted. I was getting the same in my headphones echo a minute ago. And they have the high pack up on channel 3 right now.
Yeah, the bearing to this pick site that we just want to pass over is going to be 145, and then the overall bearing is going to be more like 110. So I'm going to just, yeah, there's no ship move in, so I'll start. Okay, great.
So can you hear me now, Meredith? I'm sorry.
I can, yes.
Okay. I'm thinking they're going to downhill to one side. Is that correct right now?
Mm-hmm.
And then,
yeah, if you look at channel 3, they're showing it on the screen right now.
Oh, thank you. So my question is, I know they don't want to do fast transects, but you know how slow we've gone before. And
yeah.
So I'm kind of thinking that if we're making a slow transit, we could drop a waypoint and call it a transect, no matter how slow they're going. Because I'd like to get some quantitative info on the distribution of these sponges. I'm trying to unravel something in my head. And the other thing I know is that Abby, there's an orange sponge that looks like a fiber optic sponge without the fiber optic, without the stock.
Okay.
And a Carnus that she thinks is a new species, and it's orange. And I'm sure Abby's still on.
Yes, and I was on the dive where they collected the first one, so I know what it looks like.
And then I think the furry rather than long-haired barrel sponges, they're kind of smallish. That would be, I think, another sponge that she would like. And I will let Abby weigh in on other priorities on the science chat.
Yep. And Jenny is pointing out your mic is fading in and out. So can you make sure your mic is close?
Yeah. How about now?
That's great.
Okay. Yeah. USB-Cs and hardwired mics, not good. That's all I can say.
So front row, are you guys all caught up on the plan?
Yeah, we're all caught up. We're gonna go through this. We're gonna try to pass over this pick that we got from the multi-beam, a suspected seep.
Yep.
And then go to that 3.05. We're just gonna go real cautiously because we're going about 100 meters downslope, you know, over kind of rugged terrain, over maybe 450 meters distance. So it is off angle a little bit, but you know, we just have to be cautious as we do this.
Great.
Yeah.
Yeah.
If we could drop a marker as you start doing this, so we can have a little faux transect for 100 meters.
I'm sorry, could you say that again?
Could we drop a marker here as you head down? I know you're going slow and cautiously, but we can still get some good quantitative data there. Not a real transect, but
you want a marker indicating a transect?
Yes.
Okay, great. And Steve, do you know what transect number this is?
Oh, hi. I'm Susie, actually, this time.
Oh, hey, Susie.
Hey. No, I don't know what transect number this is.
Okay, I can...
Any folks know?
I can try to check. I have a marker, so
sure, yeah.
I'm not going to lose it. All right, I got to bridge now.
Jenny is saying that they did one transect at the very beginning.
Okay.
And this, you know, we know it's slow and it's not quite protocol, but it's better than nothing.
Yeah. So,
yeah.
Yeah. So what Bob's saying is it's going to be a little difficult to see. Yeah.
Hey, Bob, you're not on SPL, I don't think. So if you mean to be.
I know it'll be hard to see because we'll be a little higher up, but that's okay.
All right.
Yeah, so to verify if this is a transect, it would be transect number 2.
Great, thank you.
Okay, I don't know if you picked up what I was saying. Since we're going down slope, it's going to be very hard to see the bottom because, you know, the front of the vehicle's higher up than the rear. So even if I'm dragging the rear in the mud, it's going to be hard to see anything.
Yeah, because I'm looking for sponges and large sponges, I may be able to get something out of this.
Oh, cool. Okay. So you guys have been seeing a lot of big sponges on this site?
Yeah.
I think you're tugging on me a little bit, Bob.
Yeah, it's been a beautiful site with beautiful, great
so
sponges.
We're going 100 now. If you're saying 145, so 100, right? Bearing's 100?
The bearing is 100, yep. And I'm going to just keep 100 to 110.
Okay.
Yeah.
So, Jenny, do you really want us to do a transect or essentially just make time straight down the cliff to the transect and go over? Is that what you're suggesting with it's better to jump off a cliff?
Out of context.
Or is it a personal statement about
2020?
2020.
It can be two things.
Yeah.
We're moving down slope back towards this cliff feature that was covered in sponges that was seen earlier today. Right now what we're seeing is a lot of rockfish clustered around on the slope. Oh, there's a ratfish or a chimera.
It
130 now?
Yeah, I seems to be meandering a bit.
What's meandering?
I don't know. I mean, I don't know why that.
Looked like there was a lingcod that just shot across the bottom.
Still tugging on me a bit.
Yep.
Spinning.
Not out in front of you very much.
I think after we get to the seep spot, we'll go down the cliff, correct?
Yeah, so the seep spot is about 30 meters below us, and it just gets a little steeper. It's increasingly steeper as we get down. And there is a little, like a little ridge in between us and the next target. So we may be going up and down a little.
Sounds good to me.
Yeah.
Yep, that sounds great.
Okay, one more time. We're going 130?
I'm sorry, yeah, no,
Okay. One 110. All right. Yeah, it'd be way better if we weren't stepping, because every time I stop, I get covered.
yeah, and I'm gonna try to preempt him every time, but I just don't know about tracking a line.
Especially when we're dragging the rear in the mud.
Yeah. I mean, would you prefer I track a line and just monitor?
Well, are we stopping at this point?
No, I think you guys just want to visualize it, maybe confirm. I think they have the Norbit running.
I would vastly prefer keep
Yeah,
going,
let's, all right,
stepping.
let's do it.
That would be way better.
If it's better to skip this.
Oh, I think we're fine running through this pick on our way over. I think this is going to be kind of what we're seeing on the other way down.
It's going to be about 10. Let's see. It's going to be. It's going to be a good bit, so I'll just let you know. Okay. We're pretty close to that pick site, so, you know, it should be within that location. And going from the multi-beam to the seafloor, you know, there's a lot of potential for it to be a big radius around that.
Yes.
Liz and Meredith, do you want the lasers on?
Lasers on?
Yes, because we're kind of doing it as a pseudo transect, and so it'll be helpful for the folks who are trying to quantify it. Not that you can see them on the bottom right now.
Not going to be pointed at the bottom by any means.
Mm-hmm.
Okay, thanks Liz and Meredith, and thanks Bob for turning on the lasers. We're all set now.
All right, Argus, just So right behind you in the meso in
Yeah,
320. So just be careful.
What's right behind me in the meso?
I just see in the meso
because we're going down.
We're in the dirt.
We're going down.
I know. I just
my altitude's
there's a big slope behind.
I know. I just always learn to keep that out of the center ring, and there's nothing I can do about it here. Yeah, I know. All right.
Just keep marching.
Yeah. No stopping.
Looks like we have more
really
of the
down slope now.
Yep.
branch sponges.
Susie, mic check for you.
Hello?
Okay, I can hear you that time.
Yeah. So I'm just seeing some glimpses of the branch sponges, the little white branch sponges that we collected earlier on the seafloor.
Do you want me to point the camera more down at the bottom? We're going to be into the tube cores, but we're just looking out into blue water with the camera right now.
So, Abby and Meredith, would you prefer to have some tube cores and images of the bottom? Or
what do you think, Liz?
yeah, Liz, sorry.
I think the tube cores and a little, I think what we're doing right now is fine.
Yeah,
okay.
it's just...
Okay.
Okay, thanks, Liz.
So I didn't get to watch the original SpongeBob live, but I did do a little rewind, and it was quite impressive. I'm hoping we get into that again.
Yeah.
For anyone watching at home, if you're watching on the YouTube channel, you can rewind the video to watch anything you missed. I can't remember what the time interval on that rewind is. I think it's up to 12 hours. But it might not be quite that much.
So we're just transiting now to a site where there might be a seep, I think.
So we're about, within 25 meters of it. And if, it looks like we did come up this ridge, so, and the targets after this 3.05 are pretty along slope or up slope. So I'm just, if you're having trouble with the visualizing downhill, we could just like explore this area and just come off bottom and pop down to that. But that's the only thought I have about that.
Well, let's get to your spot first, and then we can kind
of
reassess and decide what to do and where to go. I suspect if we want to get back into those sponge gardens, we'll have to get on a wall a bit.
Yeah, so we're pretty much right on that pick target.
Andrew, do you want to look around a little bit?
If you guys want to.
I think if we just could sort of...
Why don't you look and see what you can find seep-wise?
Can we scan down here to the... right here?
We stopping?
Yeah, we're going to stop for a second.
And let's mark this as the end of that weird transect, okay?
Roger.
Okay, so what's the plan right here?
So can we look over just to the far left side of the screen?
Okay.
I'd like to see what's just beneath that fish, and then also at the bottom of the screen here, I'd like to see the clams that are just in front of the tube cores. See if they're clams or brachiopods.
Okay, so
I said that backwards.
Yeah, we really don't want to back up, so
If you
just plan moving ahead.
Yeah, so if you just scan
look
down
at these
right at the bottom of the screen.
Okay. Want to zoom in?
Yes, please.
Video.
Great, thank you. That's all I needed to see.
Yeah?
Yep.
Okay, zoom out.
More brachiopods.
More brachiopods, not clams. And then I'd like to go
where the fish is over there?
Where that fish is to look right here.
Okay.
We can probably zoom in from here.
Okay, zoom in, Dave.
Just right there.
Zoom in more?
Yes, please. Great. Definitely can measure that fish now.
Yeah.
Okay, we can unzoom, and if we can just sort of scan left and right.
Okay.
I think that's probably mostly all we wanted to do at the site.
Okay.
They're hidden mystery seeps.
Yeah,
Keep appearing on the sonar and then not on the bottom.
I think there's a lot of very small seeps that are adding up to ephemeral little bubble plumes that are what's confusing. And in the maps of the seeps along this region of the coast, most of them are in this 200-meter depth range. I would explain it if it's a lot of very small seeps that add up to a lot.
Abby, do you see any sponges on there that you want? Just checking with Abby, see.
Are we going to continue this direction?
That sounds good to me.
I'm putting in a step right now.
How far, did you want to go back the other way though? Or we just...
hold 1 second.
Okay, so they're not seeing any sponges that they're looking for. So I think we can continue along the path we were going.
Yep.
Thank you. Does that sound good, Liz and Meredith?
That sounds great.
Sounds good to me.
Great, thanks.
Okay,
I just want to make sure you have time to look for some seats.
So Susie, we didn't see any seeps, more brachiopods, and just sort of like the other areas.
Okay, cool. Cool.
It depends on your depth and stuff, and how much, if I'm out in front, I'm tugging on it.
It's shallow, like the pullback. Yeah. Well, I just don't want to hold it.
So we're still going 100?
Yeah, we're still going 110.
110.
Yep.
Yeah. Fragile pink urchins, rockfish, and mud.
Lots of mud.
Was it less muddy on the bottom of this feature, or pretty consistent? Guess it wasn't that big of a depth difference, 200 meters.
The top was muddy, but it was sort of hard, and then a thin layer of mud. The coring was just sort of hilarious in its attempt. So this,
yeah, this whole area looks like it has a hard with a veneer of mud on it.
Yeah.
Yeah.
And even the wall looked that way too, which is odd to see a veneer of mud on that kind of vertical face.
Nice and sticky. Trying to figure out what type of rockfish these are. I'm following along.
Do we have Erica on? Erica's still on.
I'm going to guess dark watched and find out if I'm right or wrong.
Did you want to zoom in on them, or you just want to keep the camera?
No, I'm just guessing. We can keep moving. Just careful, slow speed gives you a good chance to flip through frantically through the guidebook while we go along.
So there is this ridge.
Spine thing?
Yes, mine.
How far we got to go?
So it's still another 380 meters, 380.
Long ways.
Yep.
You doing what? 0.3?
So we're headed to that spine, is that correct? And then we'll try to head down that spine. Is that our plan, Meredith?
0.23.
Yeah, yes, that was the plan.
Very easy to see what's going on there.
I know.
Am I seeing a sponge there right in front of us? Is that one of the ones you collected already?
That little fingering sponge?
Yeah.
I think it is one of the ones that was collected already.
You want to look at it?
Did you want to go?
Oh, I think we're beyond it now, but we'll call it out earlier if we want to look.
Erica has chimed in. The rockfish we're seeing are sharp chins. Kurt as well. Pointing out to look for the black V behind their eyes. And she also confirms that they slurped one of those finger sponges earlier.
Oh, yeah.
Great.
For anyone just joining us, we're diving in Gray's Canyon off the coast of Washington State. Here to look for methane seeps and look at some pretty impressive collections of sponges that we have been exploring.
Yeah, we were first in this area in, boy, it sounds like a long time away now. What was it, 2015 maybe? And then with the Nautilus on a cruise, NA121.
That was in 2016.
There you go. And on that cruise, we saw, again, a lot of these sort of sponge gardens and we're able to take some samples. But we're going back and, oh, Eric is correcting. Now we're
fly a little lower,
trying to.
Then we can see if there's any nasty old fishing gears.
Don't want that to happen again.
It's waiting for someone to say
and it. When we get into the sponges, it is an area where you see a lot of spot prawn. So that's an area where there's likely to be a significant amount of fishing with what we call pot gear traps. But thanks to the Nautilus, on NA121, we were able to get the first samples of a lot of sponges here, some of which we think are new species. And so we're really interested in confirming those identifications. And Meredith does a lot of genetics work and start on corals and starting to do that on sponges as well.
Yep, we're trying to build what we call our voucher collection, which are individuals that have been identified species level. We then catalog a sequence for them and use them as references to compare future collections against.
And I'm coming to you from my living room, well, actually dining room. And thanks to the great efforts of the Ocean Exploration Trust and the Nautilus team, we're able to participate from home on these. And we're sorry that we aren't able to take questions from the public, which would be the normal protocol, but it's a little different time for us now. So what you're hearing is some people on the ship and some people at home. Meredith and I are at home.
Yep.
Liz Clark, I'm from the Northwest Fisheries Science Center. I'm the senior scientist there. And Meredith, I don't know, you want to go around and do some intros while we're cruising here?
Sure, sounds great. So I'm Meredith Everett. I'm also at the Northwest Fisheries Science Center, where I'm a geneticist in the Conservation Biology Department. And as was mentioned a minute ago, I do coral and sponge genetics, and I'm also doing a lot of what we call environmental DNA, which will be taking some of those Niskin water samples that we took just before the watch change. We filtered that down and collected DNA right out of the water.
Oh. It's funny how many.
I don't know, Andrew, you're on, right? Do you want to introduce yourself so I can remember what your voice sounds like?
Yeah, this is Andrew Thurber. I'm an assistant professor of oceanography and microbiology at Oregon State University. I'm the lead of the SEEP science exploration part of this cruise. It's funded through the NOAA Office of Ocean Exploration and Research. And also with me on the back row is Susie Cummings.
Yeah. Hi, everyone. I'm Susie Cummings. I am a microbiologist PhD student at OSU in Andrew's lab. And I study SEEP stuff now, but I'm interested in all sorts of different microbial ecology projects.
Looks like that's kind of bumping up. Oh, so the stutters in the video. Looks like we're coming up over this ridge.
Yeah, this is spine. Our map is pretty good.
Yeah, coming up a little bit.
Yep. So Liz and Meredith, are you going to participate in some of the other sanctuary cruises that are coming up on the Nautilus?
We are. So we'll be, we won't be broadcasting live for the next leg,